This video is about one of the fundamental principles in animal research ethics, the 3 R's concept. The 3 R's of animal research were first described by Russell and Birch in 1959. In their book, The Principles of Humane Experimental Technique, they proposed ideas to improve the treatment of laboratory animals while advancing the quality of science in animal studies. They introduced and defined the terms replacement, reduction, and refinement, now known as alternative methods for minimizing the potential for animal pain and distress in research. The first R stands for replacement. This principle encourages us to find alternatives to using animals in research whenever possible. These alternatives include mathematical models, computer simulations, in vitro biological systems, and even videotapes. The second R is reduction. It emphasizes minimizing the number of animals used in experiments. To achieve the principle of reduction, we carefully consider the species and use the minimum number of animals necessary to obtain valid results. The third R is refinement. It focuses on improving animal welfare during research. Refinement involves avoiding or minimizing unnecessary discomfort, distress, and pain experienced by animals. In replacement, methods are used that allow research experiments to be conducted without relying on animals. There are 2 replacement strategies: absolute replacement and relative replacement. Absolute replacement involves eliminating the need for animals. This can be accomplished using cell cultures or tissues, simulating biological processes using computational models, creating artificial systems for testing, such as lab-on-a-chip technology. Absolute replacement provides accurate data without animal suffering. Relative replacement also eliminates the need for animals. However, animal tissues and/or cells are used. One example of an absolute replacement is organ on a chip. With organ on a chip, cells are cultivated on a silicone chip to create miniaturized organ systems that mimics an organ's oxygen levels, nutrient gradients, biochemical cues, and other natural physiological conditions. Scientists can use these chips to study how organs function, respond to drugs, and interact with each other without using animals for testing. This technology already has applications in drug testing, disease modeling, and personalized medicine. The use of zebrafish embryos in research and testing exemplifies a relative replacement method. Zebrafish embryos are transparent during early stages of development. This allows scientists to observe internal processes, such as organ formation, blood flow, and tissue interactions, without invasive procedures. Additionally, zebrafish are prolific reproducers. They produce a tremendous number of offspring in a short time. More embryos means more experimental data. Zebrafish embryos now play a crucial role in toxicology studies. By using adult zebrafish to collect embryos, we avoid the need for more invasive procedures that directly impact the adults. In wildlife research, traditional methods often involve direct observation and physical tracking or capture, which can be invasive and time-consuming. Camera traps, thermal imaging, aerial drones, biopsy darting, environmental DNA, eDNA, marking, and footprint identification are some of the technologies that are revolutionizing how wild animals are studied. These techniques are less invasive, more efficient, and often more accurate data collection methods. Reduction. In any discussion of animal use in research, the ethical question of the number of animals required is crucial. Reduction strategies aim to minimize the number of animals used while still obtaining meaningful data. Improving experimental design and analysis can lower the number of animals needed for a given study by refining experimental design, enhancing statistical techniques, and optimizing data collection. Experimental Design Assistance (EDA) is a free online tool designed to guide researchers through experimental design, helping to ensure that they use the minimum number of animals required, use methods to reduce subjective bias, and use appropriate statistical analysis. The ARRIVE guidelines, Animal Research Reporting of In Vivo Experiments guidelines, are a checklist of information to include in publications describing animal research. Improperly reported animal research often lacks important information, limiting its utility. Reporting animal research in adherence with the ARRIVE 2.0 guidelines ensures transparent and thorough reporting with enough detail to add to the knowledge base. Reduction strategies can help strike a balance between scientific progress and ethical treatment of animals in research. Computational toxicology modeling is an excellent example of reduction. This method leverages computer simulations and data analysis to predict toxicological effects, minimizing reliance on live animal studies. It uses computational models to predict outcomes and guide the design of in vitro experiments. This helps identify potential toxic effects and refine the focus of laboratory studies, thereby reducing the number of live animal studies needed. In silico models can also predict the endpoints that would typically require live animal testing. They utilize computational data to achieve necessary endpoints without subjecting more animals to testing. Refinement is the last 3R's principle, and it focuses on modifying husbandry practices or experimental procedures to improve animal well-being and minimize or eliminate pain and distress. Better husbandry practices improve the living conditions and care of research animals. This includes the use of enhanced housing and humane handling techniques. Enhanced housing is the use of larger pens or cages and environmental enrichment. Larger housing allows for natural behaviors such as exploration, exercise, and social interaction. Environmental enrichment is adding objects and stimuli to the living environment that encourage natural behaviors. This reduces boredom and stress, promotes mental stimulation and well-being. Some examples are toys, nesting materials, climbing structures, tunnels, and when possible, food treats such as frozen fruits and even marshmallows. Humane handling should be used in all husbandry and experimental procedures. Cup handling uses cups or other gentle methods to handle small animals, especially rodents. It reduces the stress associated with traditional tail handling, leading to calmer animals and more reliable experiment results. Other gentle handling techniques include using soft gloves, offering treats, and slow, calm movements. These practices can reduce fear and anxiety, improve the human-animal bond, and ensures better cooperation from the animals during experiments. The primary goal of refinement is to ensure the highest possible standard of welfare for animals involved in research. Methods for enhancing animal well-being include regularly assessing and addressing animals' physical and psychological needs, implementing monitoring systems to quickly detect and respond to signs of pain or distress, promoting positive human-animal interactions to reduce fear and anxiety. Minimizing or eliminating pain and distress is a critical part of refinement. Methods to address pain or distress include using advanced analgesic and anesthetic protocols, applying humane endpoints where animals are removed from a study or euthanized humanely if they experience severe suffering, and continually reviewing and improving procedures based on the latest scientific and ethical standards. One key area of refinement has been eliminating food and water restriction from experimental protocols. Some techniques now used for this include using automated dispensing systems to control feeding, scheduled feeding with highly palatable substances, flavored water with highly palatable substances, using nutritionally balanced treats to avoid nutritional deficiencies, training animals to perform specific tasks or behaviors for rewards. Another example of refinement in animal research is the use of less averse methods for oral dosing. These methods reduce stress and discomfort associated with administering substances to animals. Less averse oral dosing techniques include administering liquid substances directly into the animal's mouth with a syringe, mixing substances directly into the animal's regular food, adding liquid medications or supplements to the animal's drinking water, incorporating substances into solid food forms such as biscuits or treats to encourage voluntary ingestion, and using gelatin-based formulations to encapsulate substances, making them more palatable. In summary, the principles of the 3Rs concept are replacement, the use of non-animal methods where possible; reduction, minimizing the number of animals used; and refinement, improve living conditions, use less invasive techniques, and alleviate pain and distress. The 3Rs concept guides ethical animal research practices. These methods aren't just about science, they reflect our commitment to a better world. And by prioritizing these principles, we are continually exploring innovative ways to advance science while respecting the welfare of all life forms. Ensuring animal welfare is not only about ethical obligation, but also crucial for the integrity of scientific research. Poor animal welfare can lead to unreliable experimental results, undermining the validity of the research. Animals experiencing stress or discomfort can undergo significant physiological changes, which can skew experimental outcomes, reducing the value of the data collected. The Animal Welfare Information Center, AWIC, can help by answering questions or helping with literature searches regarding the 3Rs. If you have any questions about the 3Rs or the IACUC, please contact any member of the IACUC administration team.